we will have to draw first the cycle proper. So this is your TCA. Okay, your TCA will stand for tricarboxylic acid cycle. Okay, so this is part of your glucose metabolism. So to start with, let me just give me some time to draw the cycle proper so that you could have, or I could show you the outcome of your tricarboxylic acid properly. Later I will have to explain to you what is happening in this cycle. Okay? Para makita nyo lang. Citrate. Isomerization. Iso. Citrate. AcetylCoA. And then to succinate. And then fumarate. So this is the whole cycle of your what do you call this, your TCA. Okay, so of course your intermediate that is needed is your pyruvate, and also of course the AcetylCoA. Later we will have to discuss where does this AcetylCoA came from, and then this will become your citrate, and then isomerization of citrate will become isocitrate. In this level where isocitrate will be converted to alpha ketoglutarate, there is the first production of carbon dioxide. Along with also on this level NADH will be formed. And isocitrate will become alpha ketoglutarate, and then the second product of carbon dioxide, and then succinyl CoA followed by succinate, fumarate, malate, and then oxaloacetate. Now, actually, on this part, this will play a role in gluconeogenesis. Okay? So later we will have to discuss this cycle. So since I have presented to you the cycle, the tricarboxylic acid, ito po siya, ano, Again, so this is the whole cycle of the tricarboxylic acid. Now, it is important to say what are the functions of your tricarboxylic acid. For example, in glycolysis, it has been stated that the importance of glycolysis is the production of ATP and the pyruvate. Okay? So, for the function of tricarboxylic acid, take note that your tricarboxylic acid is also known as your citric acid cycle and your Kreb cycle. Okay, so please take note that your TCA is actually the final pathway of oxidative catabolism of your fatty acids and proteins and carbohydrates. Actually, the oxidative catabolism of the said compounds converge in the tricarboxylic acid. So mamaya malalaman nyo, maintindihan nyo bakit ganoon yung pagkakasabi about your tricarboxylic acid. So take note also that your oxidative will provide your oxidation or oxidative process will provide energy for the production of the majority of the ATP. Now, please also remember that your TCA is only happening in the mitochondria. So if you're going to be mention, or if you're going to be asked where does your TCA is happening, basically this happens in the mitochondria only, ha. Mitochondria. Okay, so in the mitochondria as compared to your glycolysis, which is happening in the cytosol, di po ba? So, having it said, it has a close proximity to the electron transport system. That is why it is very effective in the creation of your ATP. Since it is its close proximity to electron transport system, make it easier to reduce NADH and FADH. Also, unlike your glycolysis can happen either aerobic or anaerobic. But your TCA is happening only in an aerobic condition. So, your TCA can only be successful in the presence of your oxygen. Now, there are some products or intermediates that are being produced during the cycle. And the tendency for those products is to pile up, naiipon sila. And you call those intermediates being collected on a certain part kasi nagpa-pile up sila. You call that intermediates as anaplerotic. So again, the piling up of your intermediates as a result of the TCA is called your anaplerotic. Okay, so what is important in the TCA is mainly the process will start on the presence of your acetyl-CoA. Okay? So, dito, sabi nga natin, yung function of your TCA, let me just enumerate it, oxidation of acetyl-CoA, and thus production of carbon dioxide. There are two carbon dioxides that are being formed. Second, for the formation of ATP, primarily by oxidative phosphorylation. Third, the GTP. And fourth, synthetic reactions. When we say synthetic reaction, it creates a component, okay, or an intermediate that can be a source of glucose kahit hindi siya carbohydrate. So, the question is, where does your acetyl-CoA came from? So, makikita nyo dyan, mayroong pumasok biglang acetyl-CoA. So, take note that the main source of your acetyl-CoA is the oxidative decarboxylation of pyruvate. So, what enzyme would help in the conversion of your pyruvate to your acetyl-CoA? So, as I have mentioned, it is because of oxidative decarboxylation of pyruvate to acetyl-CoA. Considering that it is an enzymic reaction, then therefore, we need to have a certain enzyme. And we call this enzyme as pyruvate, dali lang po dehydrogenase complex. O look at the word, it's complex. Ibig sabihin, hindi lamang po iisang enzyme yung pyruvate dehydrogenase complex. Also, please take note that your pyruvate dehydrogenase complex is not part of the TCA cycle. So, ibig sabihin, yung oxidative decarboxylation of your pyruvate to acetyl-CoA happens outside of the tricarboxylic or the Kreb cycle. Okay? So, apart from the oxidative decarboxylation of pyruvate to acetyl-CoA, take note that your pyruvate is actually the end product of aerobic glycolysis. Okay? So, bakit? Kapag anaerobic, ang end product natin ay lactate. Diba po? So, ang pyruvate ay end product of glycolysis in aerobic glycolysis. So, ito pong pyruvate which is placed in the cytosol after glycolysis simply because we know sa glycolysis is happening in the cytosol. So, yung pyruvate, nandun lang siya sa cytosol. Now, it has to enter to the mitochondrial matrix. Nandyan po. Kasi nga, ang sabi natin, your TCA happens only in the mitochondria. So, meron po tayong tinatawag na mitochondrial carrier, wherein the pyruvate will be carried and enter the TCA cycle and enter the mitochondrial membrane. Okay? So, balik po tayo dito sa pyruvate dehydrogenase complex. Pag sinabi po nating pyruvate dehydrogenase complex, this is composed of protein aggregate of multiple copies of 3 enzymes. So, ibig sabihin, ito pong pyruvate dehydrogenase complex, the one responsible for the conversion of pyruvate to acetyl-CoA, is composed of 3. So, there are 3 enzymes that is involved. So, what are these enzymes? First, your E1 or also known as pyruvate decarboxylase. Okay, E1, pyruvate decarboxylase. E2, which is your dihydrolipol transacetylase. Okay, again, E2 is your dihydrolipol transacetylase. And E3, which is your dihydrolipol dehydrogenase. Okay? Tatlo po yun ha. So, these 3 is responsible for the decarboxylation of pyruvate to acetyl-CoA. Apart from having the E1, E2, and E3, your PDHC is also a regulatory enzyme or has 2 regulatory enzymes. The other 2 regulatory enzymes being pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. Please, wag po kayong malilito. Ang responsible, inuulit ko po, ang pyruvate dehydrogenase complex, when we're talking of the conversion of pyruvate to acetyl-CoA, there are 3 enzymes that is responsible. And those are the E1, E2, and E3. Now, apart from the E1, E2, E3, the composition of your PDHC, which does not interfere or go along with the E1, E2, E3 into conversion of pyruvate to acetyl-CoA, meron pa siyang 2 regulatory enzyme. So, the regulatory enzymes that your PDHC, the complex has is your pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. Now, alam naman natin that when there is an enzyme, there should be a coenzyme. The coenzyme will definitely help in the enhancement of the reaction. So, ano po ba yung mga coenzyme na kailangan po ng DH complex o PDH complex? So, depende po yan. coenzyme will act as a carrier or oxidants of the intermediates. Okay? So, kung iyan ay iwan, ang kailangan niyan ay thiamine, pyrophosphate, ha? Thiamine, pyrophosphate. Kapag ka naman po ito'y E2, ang kailangan niya po ay lipoic acid and coenzyme A. Pagka po E3, ang kailangan niya po ay FAD and NAD. Okay, so these are the coenzymes that are needed. E1, thiamine pyrophosphate, and E2, lipoic acid and CoA, that's coenzyme A. E3, which is your FAD and NAD. Okay? So, as to regulation, dito na po papasok ang ating two regulatory enzyme, which I have mentioned earlier, the pyruvate dehydrogenase kinase and the pyruvate dehydrogenase phosphatase. The regulation, ibig sabihin, para po, ito yung stop and go, siya po yung traffic light, ano po. So, the kinase, the pyruvate dehydrogenase kinase is responsible for the inactivation of your pyruvate dehydrogenase complex. So, the activity of this PDHC is inhibited, okay? While your phosphatase, your pyruvate dehydrogenase phosphatase will activate naman the PDHC. So, si kinase, siya yung enabler. Yung huwag ka na kumilos, okay lang yan maging tamad. Okay? And si phosphatase, that is the one that pushes your PDHC na magsipag, okay, to be activated. So, take note that your kinase is allosterically activated by the presence of your ATP, AcetylCoA, and ADH. Ibig sabihin, kung mataas si kinase, hindi gagalaw ang PDHC mo. Tama? Kung mataas si kinase, maraming kinase, signal siya na, oh, you don't move. Bakit tumaas si kinase? Kasi, na-identify ni kinase, uy, marami ng energy. Marami na ring AcetylCoA na DH, so pwede nang hindi tayo kumilos niyan. Okay na. While your phosphatase, ang magti-trigger naman po dyan ay ang inyong pagbaba ng inyong ATP at pagtaas po ng ADP. So that when it is able to identify the decrease in ATP, then therefore phosphatase will ask your PDHC to react para mag-produce tayo ng more of the ATP. So therefore, if there is a high energy product, your PDHC is turned off. Syempre, marami kang ATP okay? And take note that your pyruvate is a potent inhibitor of your PDH kinase, okay? And calcium, which is a strong activator of PDH phosphatase. Again, pyruvate, potent inhibitor of kinase, and calcium, activator of your phosphatase. Okay? So, we're on the first level of the activation. Sabi nga natin, there is acetyl. So, this is the first part of the cycle, AcetylCoA production. So, pinaliwanag lang natin paano nagkakaroon ng AcetylCoA. Okay? And this will be followed by citrate synthesis. Now, the acetyl-CoA and the oxaloacetate actually, makikita nyo siya, nagsama si acetyl-CoA at saka si OAA, si oxaloacetate. Nagkaroon sila ng condensation. And this condensation is actually leading to the production of your citrate. Take note of the reaction na minamention ko. So, condensation of acetyl-CoA and oxaloacetate produces your citrate. And the condensation is catalyzed by a certain enzyme, which is your citrate synthetase. Okay? Again, the condensation is catalyzed by citrate synthetase. Now, the citrate synthetase, of course, it can be inhibited. Someone has to tell the citrate synthetase to stop. And that would be your citrate. Siyempre, pag mataas na yung content ng citrate mo, mag-i-indicate na yan na, tama na, marami na ako dito, siksikan na kami. Diba? And, your citrate, by the way, as it inhibits your citrate synthetase, it activates naman your acetyl-CoA carboxylase. Okay? And also your PFK1. Okay? So, after citrate, after the condensation of OAA and acetyl-CoA, you produce citrate. The citrate will then be isomerized to become your isocitrate. Okay? So, isocitrate. So, yung isomerization is catalyzed by aconitase, okay? Wherein there is a hydroxyl group migration. Okay? And next, after isomerization is your oxidative decarboxylation of isocitrate. Ito po, itong area na to. So, this is oxidative decarboxylation of isocitrate. So, what enzyme catalyzes the reaction is your isocitrate dehydrogenase. On this level, this is the first time that it will create a carbon dioxide. Okay? Also, the carboxylation of isocitrate, the product is your alpha ketoglutarate. And having the alpha ketoglutarate on this stage, as I have mentioned earlier, this is the first release of your carbon dioxide. Also, on this level, nagpo-produce ka na ng NADH. Okay? So, ito yan. Sa level na to, may NADH ka na. Okay? After the production of alpha ketoglutarate, there will be oxidative decarboxylation of the alpha ketoglutarate producing succinyl-CoA. So this is the second release of your carbon dioxide on this level in the decarboxylation of alpha ketoglutarate. Dun po sa succinyl-CoA, so what enzyme catalyzes the reaction? It is the alpha ketoglutarate dehydrogenase complex. Again, alpha ketoglutarate dehydrogenase complex. Same as your PDHC, pag sinabi nating complex, then therefore, this will contain three enzyme, multiple copies of the three enzyme. Okay? Now, also, same with your PDH complex. This will need your coenzyme, TPP, that is your thiamine pyrophosphate, lipoic acid, and it also needs your FAD and NAD. Okay? So, wala lang sa kanya yung CoA as compared to your PDH. So, after producing the succinyl-CoA, there will be a succinyl-CoA cleavage. So, ayun siya. Pagka na-cleave siya, magiging succinate. So, the succinate is catalyzed, or the cleavage is catalyzed by the enzyme succinate thiokinase. Okay? The succinate thiokinase, ayan po, ayan siya, cleavage, ayan. Magki-cleave siya. Cleavage, magiging succinate. The cleavage of your succinate thiokinase is actually, it cleaves off the high-energy thioester bond of succinyl-CoA. Okay? So, next reaction after the production of your succinyl-CoA. By the way, in this level, in the cleavage of your succinyl-CoA, you will be producing here, or there will be a phosphorylation of GDP to GTP. Okay? So, mamaya ha, pagka i-discuss ulit natin yan para ma-correlate natin sa function. Okay? So, phosphorylation of GDP to GTP. Okay? Okay. Then, after having your succinate, it will then undergo succinate oxidation. The oxidation is catalyzed by your succinate dehydrogenase, therefore making your succinate to become your fumarate. On this level, yung FAD mo, the FAD becomes your FADH. Okay? And take note that the succinate dehydrogenase, again, there is something special with the succinate dehydrogenase. The succinate dehydrogenase is the only enzyme embedded in the inner mitochondrial membrane. Again, the succinate dehydrogenase is the only enzyme embedded in the mitochondrial membrane. Okay? Next, you have your fumarate. Then, it will undergo fumarate hydration by the enzyme fumarase. And that will yield your malate. Lastly, you have your malate oxidation through the enzyme malate dehydrogenase, thus producing your oxaloacetate. And on this level, this is the final production of your NADH. Okay? Sa level na yan, ha? Okay, so ito yung isang function, oxidation of acetyl-CoA and production of carbon dioxide, which is shown in this figure. Now, you have your acetyl-CoA by the pyruvate dehydrogenase complex, and as we go through this cycle, citrate to isocitrate and isocitrate to alpha-ketoglutarate and alpha-ketoglutarate to produce your succinyl-CoA, there is the first and second release of carbon dioxide. So ito, check na to. So it's true. Okay? And also formation of ATP. So, saan yung formation of ATP? Take note that you have your NADH, tama po ba? Dito, no? Isocitrate to alpha-ketoglutarate. And I have forgot to mention earlier that in the cleavage of your succinyl-CoA, apart from the phosphorylation of GDP to GTP, another molecule of NADH is being formed. Okay? And then into succinate. So, this is the second NADH molecule that is being produced in the cycle. And of course, malate to oxaloacetate, no, sa hydration. So, you have your last, no, your third molecule of your NADH. We all know that your NADH and FADH also, ayan po, intermediates yan, di ba? These are the intermediates that enters the oxidative phosphorylation. And therefore, because the NADH and FADH is present, oxidative phosphorylation can happen, thus producing your ATP. Ano po? So, ayan po yung reason kung bakit nakakagawa siya ng ATP. The production of GTP as well is shown in the cleavage of succinyl-CoA to succinate. Ayan po. Phosphorylation of GDP and GTP. Okay? And of course, synthetic reactions. Paano po yung synthetic reaction? Do you remember your alpha-ketoglutarate? The alpha-ketoglutarate is actually can be from your amino acids. Okay? Not only that, your succinyl-CoA can also be from your amino acid. So, ibig sabihin, kahit hindi ka nagsimula rito, kung meron kang amino acid that will yield succinyl-CoA, meron ka pa rin succinyl-CoA. Bakit kailangan nating i-emphasize itong mga ito? Kasi, for this cycle to succeed, kailangan ang mga component na ito, alpha-ketoglutarate, succinyl-CoA, and of course, the fumarate. This is also from your amino acid, same with your oxaloacetate. So, bakit po? Because of the tricarboxylic acid cycle, apart from doing or creating your energy, your ATP, this can produce out of it the glucose. Kung mapapansin nyo, may arrow na pa ganon. So, yung oxaloacetate can be converted into glucose, thus proceeding to what we call gluconeogenesis. We produce glucose we produce glucose not from carbohydrates but from other components like your amino acids or your fats, for example. Okay? That is your synthetic reaction. That will play its role in the gluconeogenesis. So the question is, all in all, how many energy is being formed during the tricarboxylic acid cycle? So, kung tutuusin mo, meron kang, so you have your 3 NADH, which will become your 3 NAD. Okay? You have your FADH2. Isang FADH2, magiging FAD. Tapos, you have your GDP, nag-phosphorylate siya. So, ito yung energy, ATP. No? So, each molecule of your NAD produces 3 ATP. So, 3 times, 3 times 3. 9 nga. May nasi din sa 9. So, each molecule of NAD can produce 3 ATP. So, 3 times 3, that would be 9 ATP. Your FAD will produce 2. And your GTP will produce 1 ATP for a total of 12 ATPs per. Take note, this is per AcetylCoA oxidized. So, ayun kasi nga itong prima donna ng ating TCA acetyl-CoA. So, for every molecule of acetyl-CoA, you produce 12 ATPs. Compare mo naman sa glucose. For every molecule of glucose, you only produce 2 ATPs. Ito ay ilan? 12. So, ang bigat, no? So, mas maganda siya, mas mabilis siya, mas efficient siya sa paggawa ng ATPs. Now, I would like to just summarize the regulation of the tricarboxylic acid cycle, which was being also explained a while ago, no? Pinakita naman natin yan. So, yung regulation. Okay? So, take note that your TCA are the regulation. Sana maisulat ko ng marangal. Okay. Regulation of TCA. Okay? So, it could consist of 1 direct regulation and the other one is indirect regulation. Okay? So, under direct regulation, enzyme activities by product inhibition by allosteric effectors such as ATP, ADP, and NADH. So, direct regulation, we are concerned of the presence of your ATP, ADP, and NADH. Okay? So, kung meron kang ATP, ibig sabihin, matik yan, titigil yan. Kung meron kang ADP, matik yan, gagana yan. Okay? So, in the indirect, depende to sa, there are 2 types of response. Okay? There are 2 types of responses. So, una muna, if there a response to high energy state, at yung pangalawa natin ay ang low energy state. So, ayan ha, there are two type of responses, either to the high energy state and to the low energy state. So, paano ba kapag ka high energy state? Sa high energy state, mataas na rito ang ATP. So, what do you expect? Titigil siya, hindi ba? Hindi gagana ang TCA mo kasi hindi mo kailangan ng ATP dahil mataas na ang ATP mo. So, in a high energy, mataas nga energy mo eh. So, you have a high ATP. Ibig sabihin, mababa ang ADP. Remember the camp? Tandaan nyo yan. Mahihirapan kayo sa exam pag hindi nyo natandaan yan. The increase in ATP and a decrease in ADP will lead to, increase oxidative phosphorylation. Kayo na magtuloy ng spelling. And, mali. This is decreased. So, the increase in ATP, decrease in ADP, decreases your oxidative phosphorylation. And therefore, this will lead to the decrease in oxidation of NADH. Okay? So, hindi siya magiging 3-NAD. So, wala siyang mapo-form na ATP. Tama po ba? And then, because of the decrease in oxidation of NADH, there will be an increased NAD, H-NAD ratio, and this will yield into decrease or none at all the TCA cycle. And tandaan ha, high energy, marami kang ATP, mababa ADP mo. So, wala namang trigger point para ikaw ay gumana ng gumana dahil marami nga akong stock ng ATP. So, kapag ganito ang sitwasyon mo, bababa ang oxidative phosphorylation activity mo, bababa rin ang oxidation of NADH mo. Hindi mo kailangan i-oxidize ito eh. Okay? And then, therefore, there will be an increase of your NADH and NAD ratio. Now, the increase in NADH and NAD ratio will signal that, hindi mo na kailangan ng TCA. Or bagal-bagalan mo kasi nasasayang, naiipon lang. Narami naman tayo niyan. Okay? And for your low energy state, ito naman, syempre, kabaliktaran, mababa ang ATP, low energy eh. And then you have an increase of ADP. Remember, the ADP will signal the continuous production, di ba? Doon sa inyong, in-explain it din nung one time, yung camp, no? Yung acetyl, adenyl cyclase, no? Okay, so this phenomena, like your decrease in ATP and increase ADP, will lead to increase oxidative phosphorylation activity. And that will mean that you have an increased oxidation of NADH. NADH. Okay, increased oxidation of NADH, thus leading to a decreased NAD ratio. Siyempre, may signal na. Mababa na po yung NADH and NAD ratio mo. Therefore, TCA cycle activity is triggered. Okay? At the end of the day, the TCA has to act. So, ayan po yung kasaysayan ng ating regulation of TCA. Ulit, dalawa lang po yan, direct regulation. So, direct regulation will depend on the allosteric effectors. Ano po yung allosteric effectors? It could be your ATP, ADP, and NADH. Ibig sabihin, kung mataas po ang ATP, okay na, titigil na ako, hindi na ako kikilos. Kung mataas naman po ang ADP, ibig sabihin, I need to move further and produce more ATP. And the same time that there is an increase in NADH, I have to convert it, I have to oxidize it, therefore oxidation would mean creation of your ATP. Now, we also have the indirect regulation, which will depend on two type of responses, the high energy and the low energy. The high energy, ayan po ang mga kondisyon, mataas ang ATP mo, sa makatuwid, pag mataas ang ATP mo, madali lang yon, hindi na kailangan gumana ang TCA. Kung mababa naman ang ATP mo, mababa ang energy mo, therefore, kailangan gumana ni TCA. Okay? So, that's your tricarboxylic acid cycle. Actually, that's your Kreb cycle, all in all. So I hope you get to understand it well. So just remember, I just hope that you are able to take note of the enzymes that are being utilized in the cycle. And of course, hindi po mawawala ang ating activity. You have to transcribe what you have heard in this video, and you have to submit it by the end of the period. So hanggang 10:30 tayo. So by 10:30, it should be posted in the Google Classroom. Kapag ka po nagtatranscribe, hindi po tinatranscribe, hindi ko po ibig sabihin na i-transcribe nyo po yung eksaktong pagkakasabi ko. Tama naman po kayo kasi nga transcription ita-transcribe nyo kung anong sinabi ko. Pero what I am trying to extract from you is the formal transcription of this lecture, so that at the end of the lecture, you have your reviewer, and that I must be able to correct or to check your output, kung tama ba ang pagkakaintindi nyo. So, hindi nyo sasabihin lahat, pati yung sinabi kong maganda ako, nandun. So, ayun nga, masaklap pag sinasabi kong maganda ako, wala dun sa transcription ninyo. So, ibig sabihin, itatranscribe nyo siya formally. So, meaning ng oxaloacetate, malate, yun yung inaano ko, ha? So, again, thank you for the indulgence, and then I'll be just waiting for your output on or before 10:30 today. Thank you and have a nice day.